Hi. So, today I'd like to talk about DNA mutations. And DNA mutations are simply changes in the nucleotide sequence within the DNA. And so these changes are very important for the processes of life. All of our genetic variation is derived from these mutations. And so if these mutations didn't exist, we wouldn't have any kind of different traits. I mean, we would never have developed any kind of living thing in the first place. Right? So these mutations are essential to the function of life as we know it. Um, However, more often than not, a mutation in a gene is going to be harmful rather than helpful because the version of the gene that exists as it exists already works, right? And so if you start messing around with something that works, more often than not, you break it rather than make it better. Sometimes, though, you make it better. And so that's what happens with mutations, right? And we'll talk about that in the evolution unit. But for now, I just want to go into some of the actual types of mutations and what they're doing to our DNA. And so point mutations are when we're changing a single nucleotide within that DNA sequence. Right? And we have three types of point mutations. The first is an insertion, and this is when a nucleotide is added into the sequence that already exists. Right? And so none of the pieces that are already there are changed. I just take another nucleotide and it gets shoved in to the sequence that's already there. The second is a deletion. And this is when a nucleotide within that sequence is removed. I lose a nucleotide from my sequence. And the last is substitution. It's exactly what it sounds like. I have one nucleotide here at this point, and I take a different nucleotide and I swap them. So maybe instead of an A, I now have a G. Substitution. So those are my three types of point mutations for DNA. Here I have a little illustration that models how each of these mutations impact our sequence. Right? And so we have our original sequence here, ATTCGGCT. If I have an insertion mutation here between my T's, right, I've added this G. I've just plugged it in. So everything before the mutation is as it was, everything after the mutation is as it was, but I now have this extra G just kind of shoved in there. Right? For my deletion, I've deleted this second T. Right? So there used to be two T's, now one of the T's is gone. Only one T left. Right? Spilt the T somewhere. It's gone now. And then with substitution, I've taken this second T and I've swapped it with an A. Yeah? Those are my three mutations right there. Now, of the three types of mutations, insertion and deletion are definitely a lot worse than substitution. The problem with insertion and deletion is that it causes what's called frame shift. Right? And now if you think about the processes that we learned the other day with transcription and translation, right? When we get to the translation step, you should recall that the ribosome is reading the mRNA in chunks of 3 called codons. Right? And when you have an insertion or a deletion mutation anywhere in your sequence, right, your groupings of codons get messed up because you've now added something or removed something. And so wherever that mutation occurs, everywhere downstream from that mutation is now messed up. When I go to make a protein out of that information, all the coding is out of order. And so the amino acids are being selected incorrectly from that point onwards. And that's what frame shift is. It's very detrimental to the process of translation. Right? And so here I have a little model of what I'm trying to say here. We have the original set, right? So ATT, CCG, ATT, and an arbitrary A at the end. Why not? Right? And so I've chunked these into the codons, right? So my first codon is going to be coded for by ATT, my second codon by CCG, and my third codon again by ATT. So these chunks would code for the correct amino acids that would make this protein function properly. Right? Now, all I've done is plug a C in between the A and the T, right? So I have an insertion mutation here. Okay? But what that does is it messes up all of my codons there on out, right? My codon's supposed to be ATT, but now it's ACT. Right? There's a very good chance that's going to select for an entirely different amino acid. Same here. I have CCG is now TTC. TCC. I can't read the letters. Right? And then I have ATT has changed to GAT, so forth and so on. So from this point here where I put the insertion mutation in, right, all of my codons have been messed up. So this protein is just going to be wicked funky. It ain't going to work right. It's going to be completely wrong. Right? It's bad. Frame shift is really bad. Okay? That's it. Those are my 3 types of point mutations that I wanted to introduce to you today. We have insertion, where I'm adding a single nucleotide into my pre-existing sequence. I have deletion, where I'm removing one of my nucleotides from my pre-existing sequence. And I have substitution, where I'm taking one of my nucleotides from my sequence and swapping it with a different nucleotide. Yeah? Awesome. DNA mutation is done. Have a great day. Bye.